Hello, everyone, and welcome to Optimizing Expression and Purification Workflows for Membrane Proteins. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, today's event is interactive, and we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. I would now like to welcome our speakers. Alex Kirkpatrick, Field Application Scientist at Thermo Fisher Scientific, and Joanna Geddes, Product Manager, Thermo Fisher Scientific. For complete biographies on our speakers, please click the biography window on the left of your screen. Alex, you may now begin your presentation.
Thanks so much for that introduction. So here's a quick overview of what we will be covering today. First, we'll touch on some background around membrane proteins, the importance of studying membrane proteins, the challenges scientists face when looking to produce these proteins, and we'll also look at a publication as a case study for GPCR expression in mammalian cells. Then we'll dive into optimization strategies for downstream preparation of membrane proteins. And after that, I will hand it off to Joanna, who will go over some optimization strategies for some more downstream techniques. So membrane proteins are very important as they serve as gateways to cells, and they play vital roles in various cellular processes. They are a diverse group, enabling them to maintain cellular homeostasis and perform critical functions. Understanding membrane proteins is key to advancing cellular biology and developing innovative biopharmaceuticals. They are prime targets for drug development. Over 500 FDA-approved drugs target 100 unique GPCRs, 300 plus agents in clinical trials, 20% of them targeting novel GPCRs, and over 60 potentially novel GPCRs that have not been previously studied. The complexity of membrane proteins necessitates specialized expression and purification approaches. Among membrane proteins, G-coupled protein receptors, or GPCRs, stand out due to their overall variety, their 7 alpha helical transmembrane domain structure, and their significant role in mediating cellular responses to a wide range of hormones, metabolites, cytokines, and neurotransmitters. So given this central role in signal transduction, it's not surprising that GPCRs are associated with numerous disease processes, including genetic and immune system disorders and cancers. Therefore, we can appreciate why these proteins make popular targets in drug discovery efforts and as potential biomarkers. So much so that currently the FDA has approved nearly 500 drugs, as I said before, and they act on over 100 unique GPCRs. This is impressive. In order to study and understand these structures, though, for rational drug discovery, it's important to be able to produce these proteins recombinantly. However, a significant challenge in membrane protein research lies in being able to express and extract them from membranes while maintaining their structural function and integrity and stability. So as many of us know, protein production is a multi-step process, one we like to call CEPA, which stands for cloning, expression, purification, and analysis. Today we will focus on expression and purification of membrane proteins, and I'll present some process considerations that can enhance your product quality and quantity and also increase efficiency, overall increasing your likelihood of a successful outcome, reducing your time and effort spent. So the production of membrane proteins is quite challenging. Often the process suffers from lower expression yields compared to secreted proteins. Then there is the issue of the glycoprofile presence and homogeneity. So having glycans present can actually help stabilize the membrane protein. And the heterogeneity of glycan species may also be problematic for crystal formation, though, and other structural bioanalyses due to their inherent flexible nature. Membrane protein extraction, solubilization, and purification is also inherently challenging, particularly due to efficiency and stability and activity concerns. And while there is not a standardized solution to membrane protein production, there are some best practices to improve successful outcomes that hopefully help you obtain higher quantity of usable protein. Some strategies for successfully producing membrane proteins include choosing the right expression system, using stabilizing agents and optimized detergents, all which we will touch on today. So starting with membrane protein expression, there are various expression systems for recombinant protein expression. Choosing the right system for expressing membrane proteins, such as GPCRs, is not always so straightforward. Here we see the 5 major expression systems. And while bacterial and yeast systems are valuable, they are less effective in producing properly folded and post-translationally modified proteins, which is especially important in therapeutic development. And overall, these systems are less robust for producing functional membrane proteins in general. Today, mammalian and to some extent insect expression systems have become the gold standard. However, we will focus on mammalian cell expression for the duration of this presentation, as there has been a greater shift towards mammalian expression systems due to their improved protein characteristics. So mammalian systems are particularly effective for producing more complex proteins, in particular the XP293 cells, which is an HEK293 derived cell system. The benefit of these cells and the expression system is that the cells are highly transfectable, grow in suspension cell culture to high density, and with the aid of the kit reagents, they can generate superior scalable protein yields compared to traditional HEK293 cell lines or bacterial workflows. The kit format also really helps simplify the workflow by removing the need to optimize different media and reagents. The cell system is versatile in that it is capable of transiently expressing a variety of proteins, including membrane proteins. And the XP293 GNT1- cell system is particularly suitable for producing high-quality homogeneous membrane proteins, which is advantageous for structural biologists working on crystallography and NMR studies, or even those working to produce heavily glycosylated proteins, which can also be problematic. This cell line has been genetically modified to lack the N-acetyl glucosamyl transferase 1, or GNTI enzyme, resulting in simplified glycosylation patterns of the protein of interest. And it also comes in an inducible format. So here is a representative IgG protein expressed in the various cell formats, and the resulting glycoprofiles. From this, we can appreciate the simple and uniform profile achieved when expressing in XP293 GNT1- cells. These cells produce proteins with a uniform high mannose glyco pattern, as they lack the ability to add complex N-glycans. This can be advantageous for those of you who need simpler or less glycosylated proteins for your downstream analysis. Next, let's go over some work done in collaboration with a research group at NIH. This publication serves as a sort of case study to showcase the benefits and feasibility of expressing a functional GPCR to be used in NMR studies in a mammalian system, namely the XP293 and GNT1- systems. Here in this study, they assessed protein activity and thermostability. They also determined that having PTMs, or post-translational modifications, actually contributed to the stability of their purified GPCR. And ultimately, they determined that XP293 GNT1- cells are suitable for expressing their GPCR of interest, which was then used for their structural biology NMR studies. And overall, this publication lends credence to the benefit of using mammalian cell expression systems for mammalian membrane protein production. So in this study, they focused on CB2, which is a class A GPCR, as their representative membrane protein model. So let's go through some of the key data of this publication to see how mammalian expression in XP293 and the GNT1- cells could benefit your transient membrane protein production. Beyond selecting an appropriate expression system, it's important to perform optimization on the expression conditions for your protein of interest. To facilitate early-stage optimization of expression methods, a straightforward method for detection, such as GFP fusion, can be really helpful to narrow down various expression conditions. Here they utilized a C-terminal GFP his-tagged CB2 construct, which allowed for easy detection and expressed it in the GNT1- cells. And they validated it against a native and non-GFP construct, and they found that there was no significant difference between their GFP and non-GFP tagged CB2 construct in terms of CB2 expression by assessing mean fluorescence intensity, or MFI, amongst viable cells. Having validated the GFP tagged CB2 construct, the subsequent data shows how it was utilized to optimize their expression conditions. So having validated that GFP CB2 construct, they wanted to then narrow down CB2 expression conditions. So the XP293 GNT1- cells were transfected to express CB2 GFP. Then various concentrations of enhancer 1 or enhancer 2, which are core reagent components of the XP293 GNT1- system, were tested at or above percentages recommended by the user guide. Cell membrane-associated CB2 levels were then examined by flow cytometry at day 2 or day 3 post-transfection and evaluated based on MFI levels after VCD correction. So based on this, the data indicated that day 2 was optimal for harvest of CB2 in the XP293 GNT1- cells. Not only is GFP a suitable reporter to leverage during expression, but as Joanna will discuss later on, it is also very valuable for membrane protein stabilization. Next, they show that expressing CB2 in XP293 GNT1- cells in fact does produce homogeneous preparations as opposed to expression in the parental XP293 cells that express several glycosylated species of CB2. In this case, because they were expressing a membrane protein for subsequent biochemical and biophysical studies, having a more uniform glycan profile was desirable. So here they used the XP293 or the GNT1- cells to express their GPCR, and they did that as a with or without GFP fusion, and they examined the protein expression via Western blot. So from this study, they show that expressing this GPCR in the GNT1- cells, again, produces these homogeneous preps as opposed to expression in the parental cell line, which produced several glycosylated species. Retaining glycosylation and or having a more uniform glycan profile is often desirable for various subsequent biophysical and biochemical or structural biology studies. In addition, the XP293 system is scalable to meet downstream research needs. Here they conducted an activity assessment of CB2 both in membrane preparations and as purified protein by evaluating the activation of G protein in an in vitro coupled assay. When comparing the activity between CB2 sourced from a mammalian versus bacterial expression system, they found that mammalian expressed CB2 was more active and stable compared to bacterially expressed. And similarly here, they examined the thermostability of CB2 in membrane preparations and as purified protein and compared between mammalian, either the 293 parental or the GNT1- cells, and bacterially expressed protein by examining the temperature of unfolding. So aliquots were withdrawn at various time intervals and residual receptor activity analyzed by quantifying rates of G protein activation. From this, they found that mammalian expressed CB2 produces more thermostable protein than bacterially expressed. And finally, they examined the isothermal stability of purified CB2 at 15 degrees Celsius over time. And as anticipated, they found that mammalian expression of CB2 not only conferred an increased thermostability compared to expression in E. coli, but that this stability was also sustained for a longer period of time. Although we are not seeing the parallel data from CB2 expressed in the parental XP293, the results were comparable to those expressed in GNT1-. Meaning that regardless of the overall glycoprofile pattern, so whether you express it in 293 or 293 GNT1- cells, glycosylation of CB2 in general confers increased thermostability over expression in E. coli. Therefore, glycosylation overall is beneficial for your membrane protein production. However, the homogeneous glycan profile conferred through the expression in the GNT1- cell line is more suitable for structural biology efforts, as it is often the case with membrane protein production. And the increased stability and duration is particularly impactful for applications that require longer stability of protein samples, such as solution state NMR. To wrap up our discussion on membrane protein expression, let's briefly recap what was discussed. We saw how CB2 expression benefited from leveraging GFP, as well as leveraging the GNT1- cells for more homogeneous glycosylation, and that having glycosylation in general was beneficial to the proteins, all which speak to protein quality. And in terms of protein quantity, the authors report to have achieved 2 milligrams of purified CB2 per liter of culture in the XP293 GNT1- cell system. This is amongst the highest expression rate reported for recombinant full-length GPCR expressed in mammalian cells, and the first ever report of high-level expression of recombinant cannabinoid receptors in mammalian cells. Overall, this group was able to obtain high-quality, homogeneous, scalable protein preparations in a transient expression mammalian system for structural biology studies. Now let me turn it over to Joanna Geddes, who will talk about the much anticipated optimization strategies for membrane protein extraction, stabilization, and purification.
Thanks, Alex. After we've expressed our protein of interest, we then need to extract it from cells and purify it. But of course, this is a very challenging step in protein production workflow. Membrane protein complexity makes them difficult to express, and it also creates challenges in isolating membrane proteins because they're embedded in the lipid bilayer, and that creates another level of complexity when we're trying to extract. It often requires harsh conditions that result in denatured or destabilized membrane proteins, and that can cause low yields, and it requires enrichment of your target. And here we're going to discuss strategies to optimize your membrane protein preparation with ways to easily extract membrane proteins, such as GPCRs, that maintain function over time. So just like using GFP fusion protein in your expression can help you easily monitor expression levels, it can also help you easily track your protein through extraction and purification. It can also aid in solubility and stability of membrane proteins. It offers an easy visual to indicate extraction efficiency and if there's any sample loss occurring throughout the purification process. This instills confidence that you're on the right track as you move through each step and that you're minimizing your sample loss. And this can really boost your membrane protein production efforts. So whether you have a GFP fusion protein or not, it's critical to track your target protein throughout the whole process. The workflow for membrane proteins extraction and purification is notably complex, as shown here in the schematic. It's multifaceted, it typically requires multiple days, and here I have stars that indicate where attention to detail is needed to ensure that your membrane protein remains functional while achieving high purity and yield. Extraction techniques and optimizing buffer components are very crucial. When looking to extract membrane proteins, you must first decide whether a membrane prep is needed or if you can simply just do a wholesale lysis. If you have a target that's overexpressed, it may be advantageous and save time by performing a wholesale lysis, as it can eliminate many steps and reduce the time spent in the extraction process. However, if your target does not express well or is endogenously expressed, then creating a membrane prep before moving into solubilization is a good choice, as this can help enrich your target and ensure success. Efficient extraction of membrane proteins necessitates careful consideration of various factors, including the choice of reagent, buffer composition, and temperature conditions. The selected reagent must effectively solubilize the protein while preserving its native structure and functionality, and often requires extensive screening. Additionally, optimizing buffer conditions to maintain pH stability and prevent protein aggregation need to be taken into account. Lastly, temperature control during the extraction process can significantly impact yield and quality of your extracted membrane proteins, making it an essential parameter to monitor. Let's look at 3 different strategies for extraction and stabilization. Pre-formulated, detergent, and polymers have their own unique advantages and disadvantages. Let's start with pre-formulated buffers. Pre-formulated buffers are a great choice for extraction because they have been pre-optimized, which means that they already include things like salt, detergent, and additives for the best extraction efficiency. They're fast and simple with no upfront screening, and they don't require special equipment such as ultracentrifugation. It's also important to note that the particular buffer you choose may not be compatible with the downstream application when you're working with a pre-formulated buffer. In addition, because these buffers employ the use of detergents, they don't maintain that native lipid environment. So here are some examples of pre-optimized kits we offer. We have our Memper Plus kit, our GPCR extraction reagent, and then our cell surface biotinylation kit. I want to note that the GPCR extraction and stabilization reagent is often a great choice because it's suitable for downstream applications, including protein quantitation, SDS page, Western blot, binding assays, and some structural studies. It's also a very simple workflow. The Pierce GPCR extraction and stabilization reagents formulation is optimized to include detergents and stabilizers that will give the most efficient extraction of functional protein. And the data showcased on the right, the top bar graph shows improved specific binding of tritiated ligand when using the GPCR reagent compared to other commercially available reagents. In the lower bar graph, you can see that even after storing the extract for 1 week in the fridge or 1 month in the freezer, there are still elevated levels of functional activity. The basic workflow is simple. For an extraction of an overexpressed target, one can easily perform a whole cell lysis in minimal time compared to a detergent screening. The solubilization properties can also be applied to membrane preps if desired for endogenous or low abundant targets. In addition to utilizing extraction and solubilization reagents, high levels of GPCR overexpression may necessitate the use of additional mechanical forces to aid in solubilization. In this particular case, after expressing adenosine receptor type 2, various techniques such as pipetting, down homogenization, and sonication were employed to disrupt the cells and harvest the protein. The results indicated that while moderate mechanical forces like down homogenization resulted in mid-level solubilization of A2AR protein, this method was actually more effective in preserving the protein's activity compared to other techniques when we're looking at the binding activity on the right. And in addition to GPCR extraction and stabilization reagent, we offer many other optimized formulated reagents for specific sample types, as shown on this slide. Next, we have detergents. Detergents are one of the most commonly used methods. The detergent interacts with the hydrophobic region of the membrane proteins. It disrupts their association with the lipid bilayer and allows them to be solubilized in an aqueous environment. The protein is then incorporated into a detergent micelle that replaces the lipid bilayer and helps to keep that membrane protein stable. Detergents can solubilize and stabilize membrane proteins and can achieve efficient extraction. One key advantage of detergents is that because there are many to choose from, you should be able to find one that's suited for your needs. And you can even create a custom buffer formulation, but it does require some time spent pre-screening to attain efficient extraction. It's also important to note that detergents don't maintain native lipid environment. After extraction, you also may need to add additional stabilizers for your protein. Now you can narrow down your screening by choosing the optimal detergent characteristics. For example, longer chain multisides have increased solubilization efficiency, while shorter chained glucosides have increased solubilization efficiency, so they're opposite. Knowing this will be beneficial to choose one or two of each to begin your screening and focus in from there. In addition, when choosing between non-ionic and zwitterionic detergents for membrane protein extraction and stability, several factors must be considered. Non-ionic detergents such as DDM or OG are generally milder and less likely to denature proteins, and that makes them suitable for preserving activity and function. But on the other hand, zwitterionic detergents like CHAPS offer a balance between solubilization efficiency and maintaining that protein stability due to their neutral charge, which can reduce protein aggregation and improve solubility. The specific requirements of the protein, including its sensitivity to denaturation and the intended downstream application, will ultimately guide your choice for detergent. Careful consideration of several key factors can help to narrow down your screening. Compatibility with downstream application must also be ensured, as certain detergents may interfere with certain analytical techniques. Ideally, the detergent used for solubilization will be maintained throughout your purification process, and oftentimes this is not the case, and you need to do a buffer exchange step to ensure compatibility with your downstream application. Additionally, there's time and cost associated with the extraction process that are considerable considerations that you need to balance between efficiency and resourcing. We offer a variety of high quality and purity detergents specific for membrane proteins. This ensures that there is no background contaminants that may interfere with your downstream applications. Size and CMC are crucial factors that need to be considered for downstream compatibility. For example, if someone was going to purify membrane protein to go into a structural assay as in core IOEM, they're going to want the lowest CMC that's going to be efficiently extract their protein and keep it stable so that it doesn't interfere with their resolution. Depending on the efficiency of solubilization and the sensitivity of analysis method, additional exchange or removal may be required. It's important to confirm compatibility. Here are some easy to remember tips to set up an effective membrane protein extraction, as it can feel overwhelming to get started. First, it's essential to maintain constant buffer system in pH throughout the entire process to ensure stability and reproducibility. And while keeping these parameters constant, you're going to want to vary concentration and critical micelle concentration of chosen detergents to identify the optimal conditions for solubilization. Assessing the efficiency of extraction involves comparing the amount of protein solubilized in the supernatant versus what remains behind in the pellet. So you can utilize the GFP fusion tag that can significantly streamline this assessment, as the fluorescent provides a quick and reliable measure of soluble protein. Now that we've talked through a few different detergent strategies, I want to highlight one more method to consider for extraction and stabilization: polymers. As with detergents, there are different polymers to choose from, and they also require formulation and screening. But again, this allows you the flexibility to customize to your protein's need. It also, like detergents, polymers can solubilize and stabilize membrane proteins. But a key difference is that with polymers, you're able to extract the native lipids along with your membrane protein. However, due to its nature, a polymer might result in overall lower yields and might possibly interfere with your downstream applications. Common applications with polymers include bottom-up mass spectrometry, top-down mass spectrometry, lipidomics, and cross-linking. Before we conclude this section, let's talk specifically about styrene maleic acid polymers. Styrene maleic acid is an amphithetic polymer used for extraction and stabilization. It can spontaneously solubilize membrane bilayers and encapsulate membrane proteins and form what we like to call SMALPs, or styrene maleic acid lipid particles. This schematic shows how an SMA creates a lipid particle by interacting with the native lipid bilayer along with the membrane protein. These SMA copolymers spontaneously incorporate chunks of the lipid bilayer, creating these SMALPs. And they're random, so they might contain different protein-lipid complexes or just even chunks of lipids themselves. So if you have an overexpressed tag membrane protein, this method is advantageous to detergent for multiple reasons. First, it takes a typical two-step extraction and then stabilization process down to one. Second, the native lipid environment is maintained. So there's no longer that guessing of whether or not you have the right lipid ratios or the right function and stability for your membrane protein. Like detergents, polymers require screening, like I've said before. So to determine the best one for your target protein, you're going to have to want to take that into consideration. The data set here is looking at a GPCR on the top plot and an ion channel on the bottom. And you can clearly see that the different styrene maleic acid ratios will impact the solubilization efficiency based on both the target and the native lipid environment that's surrounding that target. Now, the polymer field is up and coming and has the potential to have a big impact on future studies of membrane proteins, especially in applications using mass spec, to look at structure and better understand membrane protein and lipid interactions. Also included on this slide is a database developed by Kalal Gupta's lab out of Yale University, where they're using bottom-up mass spec to better understand how different polymers enrich for different types of membrane protein targets. And this essentially is cutting down the amount of screening time that's needed for this emerging technique. Now, to summarize, a pre-formulated lysis buffer is going to give you an optimal buffering system that's going to give you the best extraction yield as well as stabilize your membrane protein of interest. A detergent is going to allow you to tailor your buffering system and the amount of detergent you add to your protein, as well as ensure compatibility with that downstream application. But it is going to require you to screen multiple different types of detergent in order to determine what is optimal. Lastly, a polymer is going to give you that native lipid environment along with your membrane protein that's going to really ensure that you have that native structure and function. It's also going to require screening, but maybe not so much as detergent, as you can utilize things like Kalal's database to help narrow down which ones you want to screen. I also wanted to show this table, which describes different techniques used for extraction and stabilization of membrane proteins. The choice of technique will vary by downstream application. If you are optimizing for a compatible detergent, it's important to also include other stabilizers like cholesterols or ligands to make sure to maintain stability. And it may be advantageous for you to take it into a liposome or a nanodisk, which would require lipid screening to maintain those functional characteristics. Now that I've talked about membrane protein extraction, I'm going to move into membrane protein purification. As we've extracted our protein of interest from our cells, now we need to purify it. So the stars here indicate the steps where buffers are crucial for maintaining membrane protein stability throughout the purification process. If we are using a detergent-based method, it's important to maintain that detergent at CMC throughout the entire purification process, so your membrane protein remains intact, stable, and functional. And if you are using a polymer, on the other hand, you don't need to add that in to your buffering system, but you do still need to make sure you're maintaining the proper temperature, pH, and salt content. Another consideration when purifying your protein of interest is scale. Understanding what your target yield is from your starting sample can help guide you in picking the correct magnetic bead or resin for your purification process. This chart captures the range of purification scales and the appropriate support to accomplish the desired purification strategy. And we have products that fit into each of these scales. In addition to different types of supports, we offer ligand-coupled supports for the most commonly used tags. And so this table over here is outlining what those ligand supports are, which tag they line up with, and what application they are most suited for. So for example, here we can see the benefits of doing a double affinity purification. In this case, we're looking at the CB2 protein that was expressed with both a His and a Strep tag. And this allowed us to do two different affinity chromatography methods to purify this protein. And when we're looking at the silver stain gel, we can appreciate how much cleaner the protein is after the second round of purification. Double affinity purification can enhance purity levels for membrane proteins. It can also facilitate buffer exchange. So in this case, we did a buffer exchange from our heaps-based extraction reagent with a larger CMC detergent into LMNG, which is a buffer with a smaller CMC, or detergent with a smaller CMC that's more amenable for cryo-EM upon the second purification. And then lastly, for the purification process, I'm not going into much detail on this, but I did also want to point out there are crucial steps in which you can lose your sample. And so those two steps are concentration as well as enzymatic cleavage. And so you're going to make sure that when you're concentrating your protein that you are equilibrating your membrane, because that will minimize the amount of membrane protein that sticks to that molecular weight cutoff membrane in the concentrator and help keep your yields high. When you're doing enzymatic cleavage, you want to make sure that you do a small scale titration of whichever enzyme you're using to cleave. So for example, TEV. You want to make sure that you titrate different levels of TEV and then look at the amount of cleavage. You want to make sure you get full cleavage of that enzyme so that you can remove that tag and then have the proper fraction, purified fraction coming off your SEC column. So here are some purification pro tips that you're going to want to take into consideration. Buffer system pH. Make sure you're holding them constant when you're doing a screening. Also, all the way through your purification process. Your detergent concentration is going to vary based on what the detergent is and what their CMC is. You want to make sure that you're holding it at CMC in all of your wash buffers throughout the purification process. And then again, using GFP Fusion is a really nice way to help assess efficiency of your purification and where you might be losing sample and may need to go back and optimize. So questions you want to ask yourself when you're setting up your purification. How much protein am I going to need at the end? How stable is my protein of interest? Are there other sensitivities I need to worry about? What's going to be compatible with my downstream application? And how pure do I need my protein to be? And if you can answer all those questions, you're going to easily be able to set up your purification strategy. You're also going to want to take into consideration what your protein source is and how that's going to line up with your ligand type. So ideally for membrane protein expression, we're working with affinity purification. We're using a recombinant tag that's expressed into our protein, and so we would choose one of the appropriate ligands here. I wanted to highlight here that we do have web pages on our website that can help you along the way. So we have the membrane protein expression and purification web page that gives nice tips and tricks, as well as products that you might want to consider when you're doing and setting up your expression, extraction, and purification methods. And then we also do have our protein extraction web page, which goes into detail on the different reagents we offer for membrane proteins. This slide is just highlighting the products we specifically talked about in this presentation. Lastly, this is a useful slide if you're looking at the full workflow from cloning, expression to purification and analysis. These helpful links take you to web pages that explain different options and products that could be useful along each step. These are our lovely field application scientists. You see Alex on here who talked before me. Please reach out to them if you have any questions. They are excellent resources and great supporters along the way. They can definitely help you out. In addition, here's some contact information for our technical sales specialist team for protein expression, so they can help get you started. Thank you all for joining us.
Alex, Joanna, thank you so much for that informative presentation. It is now time for our live Q&A portion of the webinar. Now to our audience members, if you have a question you'd like to ask, please do so now. Simply type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. Okay, we have lots of questions that have already come in, so I'm just going to dive in here. Alex, I'd like to start with this question directed at you. What about temperature shifting to enhance expression?
Thanks. That's a great question. So, of course, the general answer to that is that it's protein dependent. That in some cases, lower temperature, such as 32 degrees Celsius in a mammalian system post transfection, can help with folding. With the XP293 systems, this would be done at day 1 post transfection when you're adding the feed and enhancer. And it would be best practice, really, though, to determine this empirically for your protein of interest. That, of course, is just a general guideline, but it can be beneficial potentially for your protein of interest.
Excellent. Thank you. And Joanna, I'm going to hop over to you for this next question. How does the use of pre-formulated lysis buffers compare to custom buffer formulations in terms of efficiency and compatibility with downstream applications?
Thanks, Christy. That's a great question. The pre-formulated lysis buffer is most likely going to be the most efficient. Now, you're going to have to do screening with detergents. You could find one that's just as efficient, but it's going to take more optimization up front. But as far as compatibility, if you're moving into downstream application that you know is compatible with, it's very ideal to go with the pre-formulated. However, if you're working with a very specific protein over a long period of time, it would be more beneficial then to do those optimizations with a specific detergent and buffering system so that you get that efficiency and you can go into the downstream compatibility and it saves steps, which in the end increases your sample yield.
Thanks, Joanna. And let's stay with you on this next question. Can you explain the advantages of using SMALPs for membrane protein extraction and how it impacts the stability and functionality of the extracted proteins compared to traditional detergent-based methods?
Yeah, so when you're using a traditional detergent, the detergent itself is going to replace those lipids, and sometimes that can impact the way the membrane protein is folded. It could cause it to be tighter or looser or limit access for ligand binding. And so you want to keep that in mind because that's one of the reasons why you have to do that screening. With the SMALPs, however, you're getting those native lipids along with it. So it's really giving you that confidence that you're going to have the same functionality as you would as if it was actually in the membrane. In addition, if you're interested in the protein-lipid interactions or what that lipid environment is, you're actually going to be able to determine that information by using techniques like mass spec.
Thanks, Joanna. Alex, we'll come over to you for this next question. What is the typical harvest time for membrane proteins in the XP293 systems?
Yeah, great question. So membrane proteins would typically be harvested around about 48 to 72 hours post-transfection in this mammalian system. This time frame is really good in the sense that it balances expression levels and protein quality. Longer expression times can otherwise lead to aggregation or degradation of your membrane protein. And of course, optimal harvesting time really does ensure best practices really between yield and functionality. However, like I said before, it's always best to determine this empirically, right? So it would be a best practice for you to do a time course study on your protein of interest to determine what the optimal harvesting time would be. In some cases, it's as little as 24 hours, but we have seen some proteins go up to 72 hours. But in all, membrane proteins though do have a shorter run time.
Thanks, Alex. And let's go with this next question as well. What if I'm not looking to get a crystal? Would I still choose the GNT1 minus line?
So in this case, probably not. You could use the parental XP293 line if no crystal is needed, or if you need or don't mind more native PTMs for your protein of interest. You're still getting the full benefit of mammalian expression by using the XP293 parental cell line. The GNT1 line really just serves as a tool to address that glycosylation problem that can be troublesome for some studies.
Thank you, Alex. And it looks like we have time for one more question. Joanna, I'd like to come over to you for this one. Can you elaborate on the benefits and challenges of using double affinity purification for membrane proteins, specifically regarding purity levels and buffer exchange requirements?
Yeah, so using a double affinity exchange or a double affinity purification and an affinity purification maybe followed by a size exclusion is going to let you do like an on-column buffer exchange if you're using an SPLC for purification, which is helpful because then you don't have to do an additional step like dialysis afterwards, which can also increase sample loss. And so it's important if you need that level of purity. Now, if you don't need that level of purity for your analysis, then a single affinity purification is just fine.
Joanna, Alex, thank you so much for your time today and for sharing your expertise on efficient expression strategies. That is all the time we have for today, but I do want to thank our audience for joining us. I'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. I do want to remind our audience as well that today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's live event. Thank you again to all of you for joining us, and again to our speakers, Joanna Geddes and Alex Kirkpatrick. Have a great day, everyone. Bye-bye.